processes of transcription and translation describe the flow of information from DNA to messenger RNA, or mRNA, to a polypeptide strand. What is transcription? Transcription is the process wherein a molecule of messenger RNA, mRNA, is synthesized along a template strand of DNA. Where does transcription happen? This is a eukaryotic cell. DNA is actively transcribed in a region of the nucleus called the nucleolus. Let's look closely at a section of DNA. Part 1. Initiation. The promoter signals where transcription begins. RNA polymerase recognizes and binds to this site. The promoter is a sequence of DNA nucleotides, 15 to 300 base pairs long, that serve as the site where RNA polymerase binds. In eukaryotes, a TATA box is typically present in the promoter, consisting of a nucleotide sequence like TA, TA, A. The TATA box aids in the recognition of the promoter. First, a group of proteins called transcription factors locate and bind to the TATA box. The transcription factors are a collection of proteins that help RNA polymerase recognize and bind to the promoter. Now, RNA polymerase II is able to bind to the promoter. RNA polymerase II is the enzyme responsible for transcribing the pre-mRNA strand. Aided by transcription factors, RNA polymerase II locates the start point and begins to unwind the DNA helix. Now, RNA polymerase II can begin transcribing the DNA sequence. Part 2. Elongation. RNA polymerase II assembles the RNA nucleotides that complement the template strand. Just as in DNA replication, the enzyme reads the template strand in a 3' to 5' direction, while building the new RNA strand in a 5' to 3' direction. RNA contains the nitrogenous base uracil instead of thymine. Like thymine, it is complementary to adenine. RNA polymerase continues its path along the template as the DNA strands join back together in its wake. Let's zoom in and see this process in more detail. Like DNA polymerase, the enzyme adds nucleotides to the 3' OH group of the existing strand. A phosphate bond forms and energy is released. RNA has a ribose sugar, while DNA has a deoxyribose sugar. RNA uses the nitrogenous base uracil rather than thymine as in DNA. As RNA polymerase continues along the transcription unit, the newly synthesized RNA strand separates from the template. Part 3. Termination. As the RNA transcript nears completion, the enzyme encounters a terminator, or a sequence of nucleotides that signals the end of transcription. The RNA polymerase transcribes the terminator sequence and then continues for about 10 to 15 nucleotides before the pre-mRNA strand is released. The pre-mRNA strand, now a transcript of the DNA template's code, is released. The DNA reforms its helical structure. Before the pre-mRNA strand is ready to leave the nucleus, it must undergo additional RNA processing. Before the pre-mRNA transcript leaves the nucleus, the ends of the strand are modified to protect it from degradation in the cytoplasm. A 5' cap is added here, and the poly A tail is added here. During the early stages of transcription, the 5' end is capped with the methylated guanosine. In some cases, additional methyl groups may be added to the nucleotides at this end. The 5' cap protects the mRNA from degradation by hydrolytic enzymes as it travels through the cytoplasm. It also serves as a signal for the attachment of the ribosome. Now let's look at the 3' end. An enzyme called poly A polymerase adds a tail of 100 to 200 adenine nucleotides to the 3' end of the mRNA. The poly A tail protects the mRNA from degradation and helps in recognition by the ribosome. The next step in the preparation of the mRNA strand is the removal of non-coding segments called introns. Introns are sequences that are part of the primary transcript but do not contribute useful information for coding a protein. Exons are segments of coding sequences. The endpoints of the intron contain nucleotide sequences that are recognizable as splice sites, indicating where the strand is to be cut. An assembly called the spliceosome locates the splice sites and cuts out the intron. Within the spliceosome are smaller RNA protein complexes called SNRPs, or small nucleoribonucleoproteins. After the introns are cut out, the spliceosome joins the exons together. The intron bonds to itself and floats away. Introns are removed by multiple spliceosomes along the mRNA strand, and the leftover exons join together. Some organisms use a process called RNA editing to change the coding sequence of the transcript, resulting in new polypeptides. Now, the mRNA strand is ready to leave the nucleus and encounter a ribosome, signaling the end of RNA processing and the beginning of translation. Translation is the synthesis of a polypeptide strand using the genetic information present in the mRNA molecule. It is during translation that the nucleotide language is translated into amino acids. Let's take a look at our environment. This is a eukaryotic cell. A ribosome is an organelle comprised of protein and rRNA. Ribosomes are the physical site of translation. The mRNA transcript leaves the nucleus via the nuclear pore. Part 1. Initiation. Initiation begins when the mRNA transcript comes together with the smaller of two ribosomal subunits and a tRNA molecule. The tRNA, or transfer RNA, molecule carries the first amino acid of the polypeptide. The tRNA molecule attaches to the start codon AUG on the transcript. Now the second and larger ribosomal subunit arrives, marking the end of initiation. This ribosomal subunit has 3 tRNA binding sites called the E, P, and A sites. The incoming tRNA binds to the A site. The P site holds the tRNA that carries the growing polypeptide strand. The tRNAs will exit the molecule via the E site. Now we move into the elongation stage. Part 2, elongation. Elongation is divided into 3 steps: codon recognition, bond formation, and translocation. Every 3 nucleotides represent a codon, which codes for a particular amino acid. The incoming tRNA contains an anticodon, which complements the codon in the A site. Hydrogen bonds hold them together. The tRNA carries the next amino acid in the chain. Now the large ribosomal subunit, acting as a ribozyme, joins the 2 amino acids together with a peptide bond. The tRNAs, together with the mRNA template, move down one site. Now the used tRNA is released from the E or exit site. Then the whole process repeats. Let's jump ahead to the completion of the polypeptide. Part 3, termination. Translation is terminated when a stop codon is reached in the mRNA transcript. The release factor is a protein that recognizes and binds to the stop codon. The release factor adds a water molecule or hydrolyzes the chain, releasing the polypeptide. Once the polypeptide is released, the ribosomal subunits and other factors break apart. Let's look at the whole strand in action. Multiple ribosomes travel along the mRNA strand, simultaneously translating copies of the polypeptide. Congratulations! You've reached the end of transcription and translation, brought to you by DNA, RNA polymerase II, pre-mRNA, spliceosome, mRNA, rRNA, tRNA, amino acid, and polypeptide.